Hello friends, Janvi here. Welcome to my YouTube channel. Today I have come up with the second video on genetics. If you have not seen part 1 of the topic, you may refer to the description for the link and go through the same before proceedings with the today's topic. Before we start today's video, if you like my channel and tutorials, please subscribe to it and share it with others. Hello friends, now we are starting with the second part of genetics where we will study about the dihybrid cross, we will study about the deviation in the Mendelian law mainly called as incomplete dominance. Next we will study about non-interallelic gene interactions and different types of genetic interactions, that is nothing but the epistasis, different types of epistasis. Now we are talking about a dihybrid cross. The name says di means two. So we are taking two characters at a time to show a cross. In the dihybrid cross, we are crossing the two characters. Here I am taking an example of the color and also a seed. The color of the seed is green and yellow, whereas the shape of the seed is either it may be a round or a wrinkled. I am taking an two characters, namely the color and the seed. I am taking the two characters and we are crossing two characters with one another. So in the first parent we are considering yellow color and the round seed, whereas in another parent we are considering the green and the wrinkled seed. Let's see how a dihybrid cross will work. Okay, let's see the dihybrid cross with a diagrammatic representation. Here I am taking the two parent. First parent will be yellow color and round seed, whereas the second parent will be green color and the wrinkled seed. And the gametes here will be capital Y and capital R, whereas for the green and round the gametes will be small y and small r. When these two are crossed with each other, we'll get in a F1 generation capital Y, capital R and the small r. So total in the F1 generation we will get the plants with yellow color and the round seeds. To start with the F2 generation plants, first we have to cross the F1 generation plants. The F1 generation plants are nothing but yellow and round seeded plants. When yellow and round seeded plants are crossed with another yellow round seeded plants, the F2 generation which are produced will be 9:3:3:1. Before to start with the F2 analysis, we have to first know the gametes. The gametes which are produced by F1 generation are capital Y, capital R, capital Y, small y, capital R, and the small y and small r. These set of gametes are crossed with another set of gametes to produce the F2 generation plants. So the F2 generation plants will exist in a 9:3:3:1 ratio, where yellow and round will be of 9 and green and round will be of 3 and yellow and wrinkled will be of 3 and green and wrinkled will be in the ratio 1. Let's study about the Mendelian law of deviation. That means what? There is some deviation in the Mendelian law. What is this deviation? We will call this deviation as an incomplete dominance. We will study this incomplete dominance in the future slides, but let me give some introduction about it. Here, the Mendel took the two plants with different colored flowers, one with a red color and another one with a white color. When he crossed these plants, he got pink color for the F1 generation. surprisingly. But according to him, he has to get either red color or the white color. To study this, he further did certain cross-pollinations in the plants. So we'll check what are the things and what did he do in the furthermore slides. Incomplete dominance. In the incomplete dominance, we are taking an example of a pea plant with red color flower and white color flower. The gametes which are produced are capital R and the small r. The allelic representation will be the capital R and the capital R for red color and small r for the white color plants. The gametes produced will be capital R and the small r. So when they are crossed with each other, we will get the capital R and the small r, which is nothing but the pink colored flower. So pink color are present in the F1 generation. Now we are going through the F2 generation of an incomplete dominance. To start with an F2 generation, we have to cross the F1 generation plants. So we are taking the two F1 generation plants which are pink in color. When we cross it with each other, we will get in a F2 generation one red color flower, two pink color flower and one white color flower with the ratio 1:2:1. To see the cross for pink color, we know that is capital R and small r. When we cross it with another pink color, again which will be a capital R and the small r. So whatever we will get in the F2 generation will be both capital R or both small r, or one will be an capital R and another will be small letter r. This shows that is two will be in a homozygous condition and another two will be in a heterozygous condition. Homozygous condition will be both capital R and homozygous condition once again will be both small letters r, whereas heterozygous condition will be capital R and the small letter r, indicating the ratio will be 1:2:1. So red color flower will be 1 and pink color flower will be 2 and white color flower will be 1. So this will be the phenotypic ratio of the F2 generation of an incomplete dominance. So surprisingly, Mendel get the pink color flowers instead of getting a red color flowers in the F1 generation. This shows that there is something which is missing in the Mendelian law. So from this we will come to know that to exhibit the complete character of a particular flower, the dominancy is very important. If the dominant character is 100% is not effective, that means it cannot give up its particular character. So the dominancy can be expressed only if the 100% dominance is seen in a particular flower. Let's talk about the gene interaction. There are different types of gene interaction. Let's see what is a gene interaction. One or the two genes will interact with each other and form a gene interaction. So there are two types of gene interaction: epistasis interaction and non-epistatic interaction. The epistatic interaction is further divided into 5 types, namely a dominant epistasis, recessive epistasis, double dominant epistasis, double recessive epistasis, and double and recessive epistasis. Now we are dealing with the non-epistatic interallelic gene interactions. Here we are taking the two pair of genes which are determining a same phenotype but assort independently to produce a new phenotypes by mutual non-epistatic interactions. So when two pairs of genes interact with each other, they differ in their size and also in their shape, And now when we consider this as a non-interallelic gene interaction, I am taking an example of the combs in a fowl. When I am taking in the combs of a fowl, we are taking an example of the combs with different pattern. It may be an Vandot breed, or it may be a Brahmas breed, or it may be a Leghorns breed. But the comb will be differing in all the three breeds. In Vandot breed, comb is rose in color, whereas Brahms, the breed, will be pea in color, and Leghorns have a single comb pattern. So when we cross with each other, as we will see, there is certain deviations again from the Mendelian law. We'll see what is this later. With the help of the diagrammatic representation, I am showing how non-epistatic intra-allelic gene interaction will take place. To start with it, I am taking an rose comb and I am crossing it with a pea comb pattern. The rose comb allele is as follows: capital RR, small pp, whereas for the pea comb it will be small rr and capital PP. When these both are crossed with each other, we'll get an F1 generation with a new type of character called as walnut with capital R and small r, capital P with a small p. So there is again the deviation which is seen from the Mendelian law. That means according to the Mendel's law, we should have got either the rose comb pattern or the pea comb pattern, but for surprisingly, once again he got something different, which we will call it as a walnut with capital R, small r, capital P and small p as an F1 generation. To start with the F2 generation pattern of non-epistatic interallelic gene interaction, we are taking an F1 generation walnut and cross it with another walnut breed. To get an F2 analysis, we are getting 9:3:3:1 pattern. The walnut is as follows: is 9. 3 will be a rose pattern, another 3 will be a pea pattern, and another 1 will be a single one. Whereas the genotypic ratio of the walnut will be both the capital R and the capital P should be present to express the walnut character, whereas for rose pattern, the capital R, dominating R, should be present to express its character. To express the P pattern, the capital P, that is the dominating P, should be present. Either it may be in a homozygous condition or either it may be in a heterozygous condition, but the dominancy of the P should express in a P pattern. Whereas in single comb pattern, I will get both are in recessive condition, that is small r and small p. So the ratio will be 9:3:3:1, in which 9 will be walnut. For the walnut, you have to remember that both should be of capital R and P. Either it may be in a homozygous condition or it may be heterozygous condition. For the rose pattern, it is 3 is the one in which for the rose pattern the R should be dominating. Either it may be a homozygous or heterozygous. For the P pattern, the P should be dominating. Again, it may be a homozygous or heterozygous, but for the single, it should be both should be in a recessive condition. That means small r, small p in a homozygous condition. So, who are all not knowing about homozygous and heterozygous? For them, homozygous means both will be in an RR form. Heterozygous means one will be a capital R and one will be a small r. For your reference, I said about the homozygous and heterozygous condition. Now, first we will study with the epistatic type of gene interaction. To start with the epistatic gene interaction, first let us know the difference between the epistasis and the dominance. So first we study about the epistasis and dominance. Later we go with the gene interactions. Next thing is, what is an epistatic gene? The gene which is suppressing the other gene is called as epistatic gene. The gene which is getting suppressed is called as an hypostatic gene. Let's learn the difference between the dominance and epistasis. The dominance, only one gene is responsible for a particular character. One will be dominant and another one will be recessive. For example, if I take capital A and small a, capital A will act as a dominant one and small a will act as a recessive one. And in dominance, it is intra-allelic interaction. When we talk about the epistasis, here the two genes are responsible for a character. That means two alleles are responsible for a character, gene 1 and gene 2. And it is inter-allelic interaction. So let's learn about the epistatic type of interaction. There are different types of epistatic interaction, namely dominant epistasis, recessive epistasis, double dominant epistasis, double recessive epistasis, and last one is dominant and recessive epistasis. Let's learn in detail these different types of epistasis. Now we will start learning about an dominant type of epistasis. Here already I have said it is epistatic interaction, means two genes are present, gene 1 and gene 2. Today I am going to explain the dominant epistasis with a diagrammatic representation. So gene 1 will have an allele capital A, small a. Gene 2 has an allele with capital B and small b. So gene 1 will always suppress the character of the gene 2. That means in a dominant epistasis, the gene 1 will always suppress the character of gene 2. That means what? The gene 1 have capital A. That capital A allele will always suppress either the capital B or the small b allele which are present in the gene 2. So let me explain this with an example of the color of a flower. Normally the flower will take up the white color without any pigmentation. These genes are responsible for the pigmentation. For example, if we take like that, particular gene will be responsible for the particular pigmentation. So, for example, if we see gene 1 is responsible for the blue color pigmentation, whereas gene 1 is responsible for an yellow color pigmentation, whereas gene 2 is responsible for an blue color pigmentation. Let me make myself clear. The gene 1 is responsible for an yellow color pigmentation and gene 2 is responsible for the blue color pigmentation. When there is a cross between the yellow color and the blue color, so the parent will be yellow and blue with capital A and small b stands for an yellow color. The small a and capital B will stand for a blue color. So F1 generation will be heterozygous, capital A, small a, capital B and small b. So the F1 generation will be yellow colored flower. Okay guys, next will be, we are talking about an F2 generation. So when we talk about the F2 generation, you should be very careful about the F2 generation. So when we talk about the F2 generation, we are crossing only F1 generation parents. Okay, Let us discuss about the F2 generation in the next slide. Let's start with an dominant epistasis in which we will study about the F2 generation. So we are dealing with the F2 generation of dominant epistasis. To start with the F2 generation, we have to cross the F1 generation plants with one another. So here the F1 generation plants will be capital A, capital B, small b, and they are crossed with each other. So the gametes produced will be capital A, capital B, capital A, capital A, small a, capital B, and small a and small b. So when these gametes are crossed with each other, we will get a dihybrid cross in which the ratio will be 12:3:1. That means 12 stands for yellow color characteristic, whereas 3 stands for blue color and 1 stands for white color. So let me think, or when you think how we will get 12 colored flowers, so the explanation is here. Whatever the allele form it may be, if we get one capital letter A and other three may be whatever it may be, either it may be a small a or capital B, or small a, small b, but if one of the letter is capital A, then it always codes for an yellow color. In the same way, if one letter is capital, then it will code for a blue color. So the allelic form will be small a, capital B, or it may be small a and capital B and small b. If the blue is in capital, that means if B color is in capital, that means it will code for a blue color characteristics. And when we talk about the white color will always be in the recessive form. The recessive allelic form will be small a and small b. So you have to remember only one thing in the dominant epistasis, that is the one gene will dominate over the other gene. That means one allelic, I mean to say the gene 1 of an allelic will always dominate over the gene 2 of an allelic form. So hence we will get the phenotypic ratio as 12:3:1 in the dominant epistasis of F2 generation. Next, we will learn about another type of epistasis called as recessive epistasis. What is this recessive epistasis? Here, there is no suppression taking place between the genes. That means gene 1 is independent and it can express its character, whereas gene 2, it won't exist independently but depend upon the gene 1 for its expression. This is very, very important. Always remember, in this case, gene 2 cannot express its character until and unless the gene 1 is present with it. So the gene 2 won't exist independently and it's always dependent upon the gene 1 to express its character. So I am showing this cross with the help of the neat diagram. Please go through this. The parents here will be the capital A, small b, and they are crossed with a small a and capital B. So whenever these two are crossed, we will get the F1 generation with capital A, capital B and small b. See, there is a change here. I said capital A, capital B and small b. See here, both A gene and B gene is present and both are in dominating character. But as I said you, the B gene cannot express its character until and unless it is associated with the gene 1. So here, even though dominant A is present, it won't exhibit its character, but gene 2, which is B, the capital B which is present, it suppresses the character of the gene 1, that is the capital A. So the capital B will suppress the character of the capital A and express its character. So the gene 2 is expressing its character by dominating the B character on the gene 1, that is A. So here the character which is expressed will be a blue color 1. So the phenotypic ratio in this case will be 9:3:4. And one thing you have to remember, these are some keywords which you have to remember when you are going through the genetics. Namely, always remember, in the recessive epistasis, if both capital A and capital B is present, then it exhibits always a blue color characteristic, because here the capital B is dominating itself on the capital A and thus showing the character of the B gene. And second thing is, whenever the capital A is present and the small b is present, here the dominating will be only in A1, so it always shows an yellow colored flower. Next thing you have to remember is, this is very very important. See, for example, you will get an allelic form as small letter A, but capital B and whatever may be the next one, it may be a capital B or a small b, that doesn't matter. But remember, when the both A are in recessive condition and only one capital B is present, it won't be able to express its character. So that will always exhibit the flower color as white color. Both A won't exhibit its color nor B gene exhibits its color, so the flower color will be white in color. Whereas the next one is, both are in recessive condition. Both are in recessive condition means small a, small b. This always codes for white color. So hence the phenotypic ratio will be 9:3:4. Is it clear, guys? Now we will start with an F2 generation, but I have already explained the dihybrid cross before, so you can go through the dihybrid cross and you can check this with an recessive epistasis also. The same is done with the recessive epistasis and you will get the phenotypic ratio as 9:3:4. Just try it once and you will get the phenotypic ratio. Let's learn about double dominant epistasis. In double dominant epistasis, two alleles are present, that is nothing but gene 1 and gene 2. The allelic forms will be capital A, small a, and for gene 2, the capital B and small b. And the dominant, or I mean double dominant epistasis, both show their expression, but they will not differentiate it as blue or yellow. That means what? The pigmentation will be either of blue color or the flowers will be simply white color, because here it won't differentiate as yellow or blue. That is why means here only one allele will express its character. That means what? If capital A is present and whatever the next three allelic forms, it won't matter. It will code for a blue color flower. The same thing, if capital B is present, so only one capital B is present also, it's enough to express its character. It will express its character as blue. Only one condition will show you a white color flower, that is small a and small b. So only one recessive condition will be seen, that is of white color. So always remember here, when there is a cross which is done using a double dominant epistasis, or to explain the double dominant epistasis, we are taking the parent capital A, small b, and they are crossed with small a and capital B. So here the F1 generation will be capital A, capital B and small b. Remember, even though the A is dominant and B is dominant, here there will be no differentiation of the color. So because of the capital B and also capital A is present, it will always code for an blue color flower. So remember this one key point, only one capital A is present in the allelic form, it will show you a blue color flower. If one capital B is present in your allelic form, then also it will show you a blue color flower. In this case, it won't differentiate into yellow. So the phenotypic ratio will be 15:1. 15 blue color and 1 will be white colored flower. Now we will learn about double recessive or duplicate recessive epistasis. Here the two genes are responsible, gene A and gene B. Gene A is capital A, capital B, small b. So two genes are responsible, gene A and gene B should be present to express its character and they are codependent on each other. So the phenotypic ratio will be 9:7. 9 blue and 7 white colored flowers will be present. Always remember this keyword for this epistasis, that is when capital A and all the other three, that is small a, small b is present, it always shows white color. Next is both small a, one capital B and one small b, then also it will show white color. If both capital A and small a, capital B and small b are present, then it will show blue color. Capital A, capital B, either it may be a capital B, then it will show a blue color. Capital A, next one it may be a small a, small b and small b, it will show white colored flower. Small a, capital B and small b will show white color. That means what? If both capital A and capital B is present, only in that allelic form the blue color character is seen. That means the gene can express itself when both A and B are dominating one another. Then it will express the blue color. Otherwise, it won't express its blue color character. If any one is also recessive in this, then it won't exhibit blue color, it will exhibit only a white colored flower. So always remember, if the capital A is present and capital B is present, either it may be in a homozygous condition or it may be in a heterozygous condition, it will code for a blue color and the phenotypic ratio will be 9:7. 9 blue colored flowers and 7 white colored flower. So let's recap what we have studied in this video. First, we have studied about a dihybrid cross by taking an example of an yellow and round plant and crossed it with a green and wrinkled plant. Next, we studied the deviations in the Mendelian law, that is nothing but the incomplete dominance. Then we studied about non-interallelic interactions by taking an example. Then we have studied about the difference between the dominance and epistasis. To start learning with an epistasis, you have to first know the difference. So we have studied the difference between the dominance and epistasis. Later we have studied with the different types of epistasis. We have studied about the 5 different types of epistasis.